Shayma Glenn. So I chose to do my presentation on the regulation of sperm maturation and germinal epithelium. So our first slide here, we're going to review the anatomy quickly. And we can see here in these photos that the testes have a tough outer capsule that include masses of coiled seminiferous tubules, which make up about 80% of the testicular mass. So you can see the coiling almost looks like the small intestine, and how much of it takes up the actual testicles. The seminiferous tubules are the site of sperm production, so obviously very important. They consist of two types of cells, the actual spermatogonia that develop into sperm and supportive Sertoli cells. And the importance of these Sertoli cells are they provide nourishment for the developing spermatogonia throughout the processes. So we can see some of these, the blue cells with the encapsulated spermatogonia within inside. And then we have these Sertoli cells, are these little purple guys that we can see in this photograph. This is the seminiferous tubule kind of cut in half. We can see this outer basal layer, and then towards the center as we move inward, that's the lumen of the tubule. So moving on to the next slide, the developing spermatocytes actually stack into columns from the outer edge of the seminiferous tubule into the lumen. So we can see them kind of stacking upwards. This is a kind of a better diagram over here. And in between those are the Sertoli cells. So again, these are the ones that provide nourishment. So they're very important to the overall development of the spermatocytes. Testosterone is permeable to the basal lamina to enter the tubule. And so one function of the Sertoli cells is to regulate sperm development. And Sertoli cells are very important. They secrete many different things. So they secrete inhibin, activin, growth factors, enzymes, but more importantly, androgen binding protein. And the importance of this androgen binding protein is that it binds to testosterone, so it cannot escape the seminiferous tubule via diffusion. So as we said, the testosterone is pretty permeable to that basal lamina layer, and this ABP, or androgen binding protein, is what holds the testosterone in to help with the development of the sperm. So next slide, we kind of have an overview of the divisions that take place. So while the spermatogonia are clustered at the basal layer of the tubule, they undergo mitosis initially, which we can see in the top layer. So they're dividing or multiplying, if you will. And then some of the spermatogonia stay there and continue their production, while others begin meiosis to become primary spermatocytes. So as we move down this chart here, we can see the meiosis has the DNA replication, but there's no cell division. So we see the sister chromatids here, and that's our primary spermatocyte. From there, they go to their first meiotic division. We have the two secondary gametes, and then we have a second meiotic division, which create one of the final products of spermatids, which then give rise to the 4 sperm. So we can see there's multiple divisions going on here, whether it's mitosis or the several divisions of meiosis. It's quite a lengthy process for this entire development to occur. So for the finalization part of it, as the spermatites differentiate into sperm, they move inward towards the tubule lumen, continuously surrounded by their Sertoli cells. So they obviously, throughout the entire process, they need to be provided this nourishment. By the time the spermatocytes reach the lumen, they have divided twice and become spermatids and remain embedded in the Sertoli cells to complete transformation into sperm. So just again, reiterating how important these Sertoli cells are as they kind of surround these spermatids throughout their process to make sure they're well nourished. If not, they would very likely die off. The sperm are then released into the lumen, and so in those final processes, that's where they get their tail to be able to swim, but it does take them a bit of time to learn how to swim and learn how to finally mature, and they're not, once they're released into the lumen, they're not completely ready to be able to be on the process to fertilizing an egg. So they actually have to complete their maturation in the actual lumen of the tubule. Next, we kind of move on to the hormonal part of everything, and we see, similar to past chapters, the hypothalamus has a major part in this, as well as the anterior pituitary gland. So we can see here the hypothalamus releases the gonadotropin-releasing hormone, which it secretes, and that acts on the anterior pituitary to secrete its follicle-stimulating hormone and luteinizing hormone. So to kind of follow follicle-stimulating hormone first, this targets the Sertoli cells, as we can see down here, and they act on the Sertoli cells to synthesize paracrine molecules required for spermatogonia mitosis and spermatogenesis. So the follicle stimulating hormone also stimulates the Sertoli cells to produce that ABP for testosterone binding and retention. They also produce inhibin, as we can see over here, and this is the negative feedback loop. So if follicle stimulating hormone needs to be shut off, it actually also kind of self-regulates with producing inhibin to control its own secretions. Moving on to luteinizing hormone on the other side. This targets the interstitial cells, where testosterone is produced, and it also controls its own production with negative feedback loops as well. So we can see the testosterone here. If there's high levels of testosterone, it'll send negative feedback loops back to the hypothalamus, the gonadotropin-releasing hormone, and the luteinizing hormone to stop the oversecretion of testosterone. Testosterone is the major androgen that regulates spermatogenesis by diffusing into the Sertoli cells. They bind to the androgen receptors and initiate the functional responses required to support spermatogenesis. So we can see how this all ties in down here as the luteinizing hormone obviously activates and begins the synthesis of testosterone and increases testosterone levels within the system. And down here we have testosterone binding with APP to stay within the tubule lumen. And the androgen binding protein comes from the follicle stimulating hormone. So again, this is how all this ties into play. And then like we said earlier, when this whole process, this is the beginning part of spermatogenesis and towards the end where the sperm are actually have their tails and are beginning to swim, they still need that time to mature within the tubule lumen before they can be fully functional and fully kind of do their job of procreation process with fertilizing an egg. So again, it's a long, lengthy process. It involves hormone secretions that control what occurs, but also Sertoli cells play a major role in the overall development process of an actual sperm. That'll be it. Our sources are here. Thank you very much.